And with that, we turn to tonight's lecture on genetically engineered livestock. It's my pleasure to introduce tonight's speaker, Bill Velander. Bill is a distinguished scholar and fellow in the Department of Chemical and Biomolecular Engineering at the University of Nebraska, Lincoln. Previously, he served as chair of the department, and before joining UNL, he was a faculty member at Virginia Tech. Bill was motivated early in his career to improve the safety of the blood supply and of blood-derived medicines by the widespread contamination of blood supplies by HIV and hepatitis in the late 1980s. Since then, he and his colleagues have pioneered the production of blood proteins and other biopharmaceuticals in the milk of transgenic animals, including human protein C, anti-hemophiliac factors 8 and 9, von Willebrand's factor, and fibrinogen. He has also done pioneering work on humanizing pig tissues and organs to make them safe for human transplant therapy in place of human tissues and organs. And he has been working to develop an effective regulatory framework for these therapeutic substances and was deeply involved in developing the FDA's guidelines for therapeutics derived from transgenic animals and plants. Bill is an author on numerous scientific publications, and he is an inventor on a variety of patent applications and patents relating to blood proteins and transgenic animals. In fact, Bill and I first met about 25 years ago, working on these patents when I was just beginning my career as a patent attorney. His work has been featured in the popular media, including in a cover story in National Geographic, articles in Scientific American, Smithsonian Magazine, and Discover Magazine, a broadcast on the Discovery Health Channel, and an exhibit at the Chicago Museum of Science and Technology. He also served as science advisor to Harrison Ford on the film Extraordinary Measures, about the contributions that scientists have made to medical care. He is an elected fellow of the American Institute of Medical and Biological Engineering, and he served as AIMBE's ambassador to Capitol Hill. He earned a BS in biochemistry at Illinois' Benedictine College, a master's of chemical engineering at Illinois Institute of Technology, and a PhD in chemical engineering at Pennsylvania State University. His talk tonight is entitled Genetically Engineering Livestock, Bringing 21st Century Medicines to the Developing World. Please hold questions to the end and join me in welcoming Bill to the podium. Bill?
I'm just honored for the invitation to talk about my research, and really, throughout this talk, I have certain citations which indicate how big this group was, and to a certain extent, how difficult it is to tackle these kinds of problems unless you have a team full of really bright, accomplished people. And giving this talk in Washington, actually, to this society is pretty apropos, because all of the training we did to be able to do this kind of work was due to the people from the Washington area at NIH and the Bethesda campus, and at USDA at Beltsville, and most of all, and you'll see a lot of names in there that I'll refer to, from the American Red Cross Jerome Holland Lab in Rockville. So I don't want to underpraise them. They were just, all of these people were just the keys to putting a dent in this problem, and hopefully a solution to some really important health problems that deny perhaps 80% of the world from receiving quality health care like we do have in the United States and in Europe. So with that, I'll begin, and I'll be really happy to answer questions at the end, and so please don't be bashful with that. So coming here and having such nice weather is such a pleasure. We miss that of Virginia. Blacksburg is a very temperate place with very little winter compared to Nebraska. But Nebraska has a lot of very attractive things, in the sense that I do work with livestock, and 1 out of every 5 steaks comes from Nebraska. So So that actually is a good thing, and we have some things in touch besides some good quality medicine at the University of Nebraska's Medical Center, which was one of the reasons that led me out there. The other things are just salt of the earth people, which I've always been taught, bloom where you plant, where you're planted, and I can say Nebraska, although I didn't anticipate it is that it would reach these kind of heights, but it truly is a good place to bloom, and don't want to understate that. It has some really unique features, like its third largest city is the Memorial Stadium, with the home of the Huskers, on a home game in the fall. It is a harsh place in the wintertime, and our students, I give them lots of kudos for being able to study after being so frozen. It's not so hard on the professors. They really make us, they adapt our life so that we can get to class on time and do justice to that. And although we didn't quite hit the cherry blossoms like we should, we will have some, not as good as the cherry blossoms, but something that will greet us in about a month in Lincoln. So again, what I'm talking about today is a lot of biotechnology in really some of the most complicated proteins known to man, and therefore the most complicated molecules known to man, and molecules that even your own liver has a problem making with any efficiency. And so that says something. And what we're trying to do is take advantage of that infrastructure that does a better job than most of the rest of the animal world at doing very complex biochemistry to make a very functionally complex, structurally complex protein molecule. And I want you to see that. And these collaborations, this slide is actually for two reasons. The collaborations are broad and include University of Michigan, and a laboratory owned by the French government, which used to be owned by Genzyme, called Genzyme Transgenics, but is now owned by the French government to make transgenic animals, and they have an all-star cast of scientists there. People of course, I mentioned NIH and USDA, and the Jerome Holland Lab has been tremendous at UNC Chapel Hill. And in addition to the fact, UCLA and UC San Diego. So there's a lot of important places. And of course, in Europe, we have some in touch. I mentioned the French government owning this transgenic entity in Boston, but also now with Brazil, and I'll tell you a little bit about that later. What's very stark about this, and the major theme of this talk, is that you see there's no stars in the rest of the world, and that really here is to indicate that's where the majority of the population on the planet is, and they don't get access to the kind of health care in really tragic diseases that could be well treated by biopharmaceuticals, complex protein molecules that are available to us, but not to them. And so in the future, we'd like to see stars there indicating that they have access to this kind of technology and therefore medical care. This is a very profound slide, and I felt that it expresses a really important point, and that is this hemophilia type B patient, his name is Michael, is a Cuban boy. And this is at a Cuban pediatric ward in Havana. The lady next to him is Dr. Carol Casper. She was arguably the first one to treat hemophilic patients with factor. You had another one of the first people to treat hemophilic here in your area, Lee Hoyer at the American Red Cross, for treating hemophilia A and hemophilia B. And we're going to talk about that in a more detailed way. Michael can't put his leg down because he has a serious bleed in his hip. And the reason why that happens with too great a frequency that will lead to such profound degradation of Michael's joints by the time he is 40, if he's still alive. He'll be nearly walking on crutches most of the time because of arthropathy that is destroying his joints due to bleeding in the joints, because he's not getting factor IX, because that kind of therapy would cost $50,000 to $100,000 a year. And that's not something that can happen in a developing country, even with excellent health care like Cuba has. They can diagnose, but they can't treat. And this is a problem. The ability to treat diseases like this relies on your ability to get these biopharmaceuticals. And I want to add that, and this is a very important thing, that doctors like Carol Casper, who have dedicated their lives to try and improve the situation in developing countries, has not only spent her life training people on how to recognize this disease, to diagnose this disease, but she spends a lot of time on airplanes bringing donations of factor VIII and factor IX to these people to save mostly people that are undergoing surgery. It's not a lot of material, but it does do something to keep hope in the physicians that are treating these kids in all these countries. So India, Latin America, even China recently, up until recently. So this is a very profound mission that these people are on, and I was so happy to be mentored by these people and to be shown that having something to be dedicated to in life that is this profound is just one of the most beautiful lessons ever to learn. One of the things I want to talk about is cost, again, and the reason why that cost exists. It's very variable. So an average cost for a patient is difficult to pin down because of the profound complexities that the most severe of these patients get, which sometimes end up costing, in the United States, $400,000, $500,000, $900,000 expended over 6 to 9 months to conquer adverse immune reactions to a factor that they've been given that their body doesn't recognize because they don't make it. So this is a profound thing about biopharmaceuticals. They're proteins. The immune system has a very strong reaction to the surveillance that it has for proteins. And that's one of the most serious things for biopharmaceuticals in general, is that immune reaction, which requires yet another level of technology to complement the accessibility to these biopharmaceuticals to at least initially treat the diseases. But the bottom line is that this is too expensive for 80% of the world to be able to do anything but even barely diagnose, but certainly not to treat. The proteins we're talking about here and in tonight's lecture is, if I can, oops. If I could, no, I can't get that. I'm gonna ask you to beg for forgiveness here because I can't operate the laser here. But the point is that factor VIII and factor IX are at the core of the pinch point for generating the biggest wave of procoagulant activated enzymes in the coagulation cascade. And you can see this cascade up here. It starts up with 2 different activation points for 2 different reasons that require clotting. But one activates 10, 10 activates 1,000, 1,000 activates 100,000, and so on, until you get this tidal wave of coagulation activity that ends up catalyzing a tidal wave of thrombin and then a polymerization of your fibrin to form a hemostatic barrier that is covalently anchored to a wound site. So we're going to talk about that important pinch point, and that's factor IX and factor VIII. Without a natively active factor VIII or factor IX, you will have almost identical bleeding diathesis, and that is really only curable by having a replacement factor VIII or factor IX. We won't talk about gene therapy tonight. There are still problems with that, but there is hope for that. I don't know what the economics are going to be. That's a complex question. I work with a lot of gene therapists because I am pro-therapy no matter what. But we will focus tonight not on gene therapy, but on simply replacement therapy, which will be at the first level of quality care, an IV, an intravenous therapy. If we can make enough of this, we can make it a non-IV therapy, maybe buccally, maybe orally. And that's a very, very fascinating thing. It's hard to predict sometimes when you have a new technology where it can take you, if it can take you in really unimaginable ways, in this case, unimaginable volumes of complex biotherapeutics. So we're going to talk about factor VIII and factor IX and fibrinogen, who are key players in the coagulation cascade. Factor VIII and factor IX are X-linked diseases. And the thing about X chromosomes, for those that are not used to thinking in that way, is that if you have normal levels of factor VIII or factor IX, for example, and you have a defect in one of your alleles, and that allele can't be countered by a backup copy, which would be on your Y chromosome, so you don't have that backup copy, you will have 50%. If you have a good copy on your X, you'll have 50% of the levels. You won't have any real bleeding symptoms. But if you have a defective copy on your X, and then you're in trouble. Then you have a profound bleeding problem. So it's an X-linked disorder. And it's really, I think, let's see here. It's really errant. It's really errant to say carrier. It's not when they say, well, the mother was a carrier. That's a convenience, but it's scientifically that person has reduced factor VIII and factor IX. It's just that they can get away with it under most circumstances. If you get into certain adverse health situations, that 44% represented by the X with the defect, that's red with the normal X, that may not be enough. That levels of 44% may go down to something like 10% momentarily, in which case you will have some bleeding problems. So carrier is not quite the most accurate term. And I want to add that over the generations, you can see levels spontaneously fall off a cliff, and there's a reason for that, is that the complexity of the proteins actually can be traced back to the complexity of the genes and the size of these genes, the combined complexity and size. And so the mutation rates are extraordinary because of the fragility of these genes. And that is why you see a 1% level, which would be severe hemophilia, in this person that inherited originally from a grandfather that had almost normal levels of in this case, factor IX. That size of those genes, factor VIII is a really big one. It's 105 kilobase pairs. So you're not going to do a whole lot with that in terms of fixing it en route, although we talked about that this evening, and I was intrigued about changing things at an RNA repair level. But both factor VIII and factor IX have 1 out of 5,000 males born and 1 out of 23,000 males born, respectively. Look at the size of the genes. The amount of DNA there and its fragility is related by how much of gene has to be kept intact before you get an adverse result like a bleeding diathesis. And the other thing I think is very profound, and so when we have this therapy in the United States and in Europe and in Japan, for example, we say we have ours. This is really great. We've been able to take care of it. But this disease occurs at the same frequency in all peoples. So that same rate of mutations, even if somehow you could not have any hemophilia people, the next year you'd have the same frequency of people being born because those mutations happen due to the fragility and the size of these genes. That happens in the rest of the world. So they have a tremendous problem. They will have this same problem with lack of treatment forever, no matter if their hemophilic kids die of the disease. Most of that care, and this has been tracked in the most developed countries where there is care available. You can see the thousands of dollars that are spent on severe hemophilia, and if they develop immune inhibitors, antibodies against factor VIII or factor IX, you see that the costs are quite astronomical, and it is a really very, very tough treatment for 6 to 9 months with a port to have continuous daily infusions, which are can be. I know many cases that were $1 million, not just 400,000. Again, most of the cost is even if you try to do prophylactic care, which requires a dosing due to short half-lives, which is another part of what we'll talk about. Prophylactic IV care, meaning you're getting infusions, IV infusions every 3 days. That's what seems to be a good target for preventative care for gene destruction. Joint destruction. The vast majority is always not the caregiver, but the factor itself that you're infusing. The factor IX gene, if we just focus on that for just a little bit, and then we talked about it's not a small gene, it's not a huge gene, but it is fragile in a certain way that really, if you get these mutations, you will have a defective phenotype, a bleeding diaphysis. And so the molecule itself has a lot of very complex domains. And most people that are familiar with the complexity of the most complex proteins would say that this kind of protein it's in the top 5, if not the top 2 most complicated proteins. And if we look at it, a lot of it's due to post-translational modification. So it's translated into a single chain, and then a lot of different modification happens on that protein backbone, which is essential for 2 different things. How it can recognize that a clot needs to be catalyzed at a wound site. How we can recognize that wound site exists, the post-translational modifications allow it to do that. It allows and enables that structure that allows it to do that. The second thing about the post-translational modification is it allows its residence time in plasma, mostly independent of its enzymatic activity. So the pharmacokinetics and its ability to initiate the clot are mostly independent things, and that's part of the complex structure function of this kind of a protein, and it's a vitamin K-dependent protein. It has gamma carboxylation, which is absolutely needed for both the pharmacokinetic behavior to be relatively long and also the enzymatic part of its activity, which creates the clot. This post-translational modification is diverse. It includes phosphorylation and sulfation, as well as glycosylation in 2 different major forms of glycosylation. It is heavily disulfide bridged. There's the gamma carboxylation, which is a difficult biochemistry for even animal cells to do. And it has to have a propeptide removed, which is another modification that has to happen through its route from the endoplasmic reticulum into the Golgi and then secreted out of the cells. And we'll talk about that a little bit. So that whole concept of complex proteins being delivered to where they have to be in a steady state way so that you're always covered against any clotting that has to happen at a wound site, you're always covered for that. That's a very complex phenomena, and we're just now, in the last 5 to 8 years, understanding that what we measure in blood actually doesn't correlate necessarily as well as we once thought with trying to prevent bleeding in this case. So a lot of different coagulation proteins are now, we're finding that out, that their best place to reside, hindsight is 2020, is where the clotting has to occur, at the vascular surface, if that's disrupted. So it turns out with factor IX that there's 10-fold more factor IX, this is just recently discovered, on your blood vessel walls than there is circulating in your blood. That makes a difference because we don't treat hemophilia B except by the levels we have in our blood, and it is a complex enough phenomena in terms of that equilibrium that may not be the best rule of thumb. And if we stain the endothelium, the surfaces of our blood vessels, this was done in a mouse, and there's something profound about this. Two things. One, we have just barely enough material to really elucidate this in a mouse. And a mouse is not that close to us. So if we're going to make the quality of therapy go up, we have to have better understanding, but that means we have to do larger animals, but it means we have to have a lot more material to really find out what's going on in our bodies. That's a profound problem with complex proteins, particularly that have to do with coagulation. One of the things we're going to talk about is that this ability to partition into the reservoir is actually due to a very complex function and structure that comes from the vitamin K-dependent modification called gamma carboxylation. And a very subtle perturbation of this thing, the omega loop, affects the way it partitions into this extravascular space. We'll talk about that more later. But you see, there's an awful lot to do, and you're just starting the race. You're just getting to the starting line when you have enough material to start seeing how does that kind of structure affect how it's going to partition into my blood vessel surface, and therefore, how am I really going to understand what's going on with this disease and treat it in a much better way? And even with people with prophylaxis, still end up having, age 50, some disturbing arthropathy. That's still happening. So prophylaxis is not the end all. We still need to understand the disease physiology, pathology, but we're not going to do that until we have lots of material to do large animals like dogs and pigs. Okay, so we need abundant factor IX or factor VIII, the other coagulation factor, which we'll talk about a little bit. It's a lot harder, a lot bigger beast than factor IX is. We need to do the large animal studies. We need to increase our knowledge of the physiology of these proteins and at the blood vessel surface. And then we improve the facsimiles we have, because anytime you make recombinant biopharmaceuticals, you're making a facsimile, not a Xerox copy. Something that sort of looks like what your liver makes. And how does that affect the physiology? We don't know until we iterate in this cycle that I put up there. And before we can do that, we have to redesign the molecule, maybe. Maybe to get, if we have the proper understanding, to get the dramatically improved therapeutic result, we have to redesign the molecules so that we fit all of this together in one logical progression that ends with combining all of it and making the quality of life better for the patient. So we're just at the first level in the United States, is with as much as we've invested, and we'll talk about that investment in a second, we're still only at the first level of care, not the highest quality we can get. That's a stark contrast, though, to the developing world. 80% of the population gets nothing. Where's the cost? Why can't we just share this with everybody? for factor 9 or factor 8, the factories made in the last 8 years, for recombinant factor 8 and recombinant factor 9, it cost $1 billion of upfront capital investment to make one factory that services just us and Europe. There is no way anywhere else in the world can afford that kind of upfront investment for today's production technology. That was a very astonishing thing and a very discouraging thing. But when you look at it and you look at how much material has to be produced, and that's a rate process. That's a milligram of that's like a milligram of protein per hour per liter of reactor fluid sort of analysis. But when you get down to it, the low cell density that you have in bioreactors, classical cell culture bioreactors, you see that there's not enough cells in there. You can't get the cell concentration up high enough to make a lot of material to conquer the problems, even at the research level. Just have an abundant amount of factor 8 and factor 9 to do the large animal studies that you need. You need to be able to throw buckets away of it just to find out the way the disease really works. So how do you do that? Well, you need something like 100-fold greater productivity, which means you need a lot of cells in that bioreactor relative to the fluid that they're secreting factor 8 or factor 9 into. How are you going to do that? This talk is about using the mammary gland, which does a really good job. It does a really good job of doing that because it has 10 to the 9th cells per milliliter of milk that it's secreting in because of the structure of the tissue. Our current bioreactors, when you install them into a $1 billion upfront investment in a factory, have only 5 times 10 to the 6th ml cells per milliliter of bioreactor fluid. That's orders of magnitude off of where you need to be. But that's what we have right now. So can we take advantage of that structure? And the high cell, this is a histology of a cross-section of a mammary gland that's lactating. And this is from a pig. But that's how we get that 10 to the 9th cells. That's how you can visualize the 10 to the 9th cells per ml and the kind of productivity that could give you if you could genetically engineer those cells to make the protein that you want, and if those cells have the biochemistry to do all that post-translational modification I just talked about. Lots of stuff has to happen, but you have a shot at the productivity part in terms of milligrams of protein per hour per milliliter or liter of fluid that bioreactor is going to produce in milk. And so you got 2 different kinds of bioreactors you have to do research on. This was a very, very nice article as a primer. It was our first step in fulfilling this dream. And this is I'm particularly proud of this one because 2 of my collaborators, and this is kind of a profound thing for people that are scientists, these are tough problems, and even when you have a team of people who are dedicated and really smart, hardworking people, like Henrik Luban and Bill Drohan of the American Red Cross up in Holland Lab. This project started in 1987. Both those guys have passed away since then. This is profound. When people talk about investing in science and technology, you have to be in it for the long run, and that means longer than the lifespan of a lot of scientists. That's a profound statement. Didn't we just give you money last year? If you want an answer, just a dent in the problem, you're talking about a lifetime of research. So hard to put that into what that's going to cost, but it takes some dedication from society to make sure that happens. So every time NIH budget gets threatened and not expanding, after you adjust for inflation especially, it makes a guy like me nervous. 2 of my dear compadres are not here anymore. They didn't see the finish line. So we're talking about animal cells in the bioreactor. The $1 billion factory is animal cells. We're talking about genetically engineering animal cells, but in the context of a tissue, of a living animal, to make really complex proteins like factor IX. When we first started this, we learned at NIH how to make a transgenic mouse by pronuclear injection. We learned how to make a transgenic pig by pronuclear injection at USDA Beltsville. That was a tremendous kindness that they, because they weren't doing our research, but they had a reason to do transgenic animals, and they were pioneers in it, and they took pity on myself, our grad students, other scientists at Red Cross also that didn't know how to do this. We were able to do it. It was good for the time, but when you rely on the natural maintenance biochemistry for inserting genes, your foreign gene, in this case for factor IX or for factor VIII, going into the chromosomal makeup of the pronuclear stage of a pig embryo. That's a pig embryo. I can tell because it's been centrifuged and all the lipid in the embryo is pushed to one side so that you can see the pronucleus of the embryo. And the female pronucleus is the one that you can actually inject into. And then, again, after that's injected, about 1,000 copies of the gene, you rely on the natural biochemistry to insert it. That's a random process. You end up with a lot of mosaic animals because the integration maybe doesn't occur in the first cellular mitosis. So you end up with some of the cells of the pig having it, not all of them. Not very efficient, but we were able to generate enough pigs, maybe of factor IX, our first 20 lineages. We had 3 or 4 that were, the insertion happened before the cell division. Before that chromosomal duplication. Since then, we use cloning. Cloning has a tremendous advantage that you do it in the petri dish and you look to make sure of propagated cells that you've transfected, and that's by the green cells up there, that you select for them with selectable markers so that every cell eventually in your series of culture experiments have the genes. So you don't worry about this mosaicism problem. Then you culture those that all have the genes, and you can genotype those too. You can genotype that culture and say, I like the number of copies in this one, and I think that's going to be more stable. You take the nucleus out of a freshly fertilized, in vitro fertilized embryo, you remove its nucleus. You saw in a previous slide with pronuclear injection, you're actually injecting into it. In this case, we take a similar pipette and pull the nucleus out, and then we take the nucleus out of the cultured cells and we replace that one that we took out. And that's called somatic cell nuclear transfer because these cells come from somatic cells, like skin cells. Dolly the sheep came from skin cells on her mammary gland. We'll talk about that a little bit later. But this is a very efficient way because you know that every embryo born from that nuclear transfer is going to be transgenic. That's a big improvement, and there are other things that are going to happen to make that even better, and we'll talk about that towards the end of the talk, but this was a big thing for us. Here's that process of pulling the nucleus out and then reinserting the one that we know has the integrated transgene. In this case, this was for factor IX. You culture those embryos, and when you know you've got a nice, healthy embryo, because the physical nature, the mechanical nature of this kind of process really is hard on most of the cells, and most of them do not divide and get up to blastocyst stage. And so you only pick the ones that look really healthy, and you can see just a couple of really healthy ones up there, and a lot of degenerate ones. But you have to transfer 125 of those to get a pig pregnant, and that takes a lot of work in the lab. So when I say we had a big team of skilled people, there's a lot of skill in doing this. So it was a lot of contributing scientists to this in the lab, and skilled technicians, skilled grad students, a lot of people. So it looks simple up here, but it's not that simple. Okay, so Why did we choose pigs? Well, this was one of our early experiments. We felt like we had to outdo Dolly the sheep. They were one of our collaborators, and we just felt really embarrassed that we didn't come up with it. So we came up with this. This is a high growth rate, no waste pig. Smithfield wanted this one because they felt it might be a product advantage for making hams. But truthfully, we chose the pig because they are easier to genetically engineer, but they have a short generation time and a short gestation time, and they have lots of piglet data points that we can study. The average pregnancy, if it's to hold, has to be 4 to 5 piglets. But a healthy pregnancy could be up to a dozen. And so when you're looking at having to study a lot of animals to decide what kind of biosynthesis of these proteins like factor VIII and factor IX are going to happen in the milk a year later after the litter is born, that's an advantage. Only a year later, and I've got milk, big advantage to pigs. This is one of the reasons we chose them. But perhaps the most astonishing part of it was, does the pig have the biochemistry to do that really complex post-translation in translational processing in a reasonable facsimile way so that it could make factor VIII or factor IX one, number 2, one, number 3 most complicated molecules known to man? If you're going to get a stopgap organ transplant in the future, you're going to get it from a pig, a genetically engineered pig. Why? Because their biochemistry is so close to ours. What has to be altered is their immune system that has to be humanized. But that similarity in the biochemistry, that profound kind of reference about how similar they are to us, I mean, it's true. They do love beer. They do love potato chips. They're our best producers. They can have that. But the more important thing is that they're very similar to us in biochemistry, and it turns out that their mammary gland is too. Very similar to human liver biochemistry. And the other thing is, I have to give a plug for my wife here. My lovely bride. She works for a lactation clinic that helps so many young women that are in a fragile state with a new baby, a lot of them single parents with not a lot of support, but lactation brings those kids back from the precipice and into a reasonable early childhood. And so having research with lactation actually has made a lot of good bridges with my wife and I. We really do well with that. So I just wanted to give her a shout out for that, because that's an important service to society, more so than any lactation work that I do. So now we can clone. We can guarantee that our manipulations are going to come out with the genetic engineering that we need. So now it's time to improve the post-translational modification, take a step up. We look at the baseline cases. It makes pretty good factor IX. It still has the propeptide on it. That's a complexity if you're trying to purify it. Propeptide factor IX, containing factor IX, and mature factor IX that does not have the propeptide. It's a little hard to purify that, and you don't see much of that in your bloodstream, so we always wonder if we have a little contaminating factor IX and it goes into a human being, will that create an immune problem? And so we'd rather not have it there. So how do we improve that? That's a difficult thing to do, to chop that propeptide off. So we've devised a way to genetically engineer the pig, and with post-translational processing, with the gene that actually is responsible for making the protein, which is called furin, that clips the propeptide off. Now, that's a kind of a substrate to enzyme ratio thing. You don't need a lot of furin because it's the enzyme. The substrate is the factor IX. How do you do that? You adjust the structures of the genes so that, in this case, the promoter was shorter for directing the synthesis of a protein, a transgenic protein that's not normally there, to tone it down, to just make some. Use a shorter promoter to do that. The longer promoter is to drive it as hard as we can so we make as much factor IX as we can. Say a substrate factor IX to furin ratio, in this particular case, to get the propeptide chopped off, maybe 1 to 100, 1 to 200, something like that. But doing it at the gene control level. So we developed those two genes, and we put those into the pigs by cloning. I want to add that I don't have a shout-out slide for all the incredible collaborators I have, but every time I show a lot of names, for example, this last paper, there's 17 authors on that to do this job. It took 4 years. About half of our NIH grant, about $5 million. So this is not trivial stuff in terms of getting this done. You need a lot of skilled people, and every one of these people did that. And I would invite you to look at that paper and see this, because it really is, I think, a tribute to a big team of people being able to work and get something done. So when we did this and we looked at the top is the anti-factor IX Western blot, so you can see that all the pigs we're talking about, and we have 5 different lanes there, and so if I look at the kinds of pigs that are up there, first we call them monogenic. They just have factor IX, so they're just making factor IX. They're going to have the propeptide on the end of the factor IX in a pretty large percentage. And then we have pigs that have the 2 genes. We call those bigenic. And then we see how does it make the processing enzyme at a level that'll take off the propeptide. And what we see is that, sure enough, the pigs, if you look at the anti-furin panel, only the bigenic pigs are making the furin. All the pigs make about the same level of factor IX. They're under the same genetic control. So it's a pretty reproducible bioreactor from that kind of product synthesis control point of view. And then you see, of course, those are the first 2 pigs that don't have the furin. They have the endogenous inability to clip off all the propeptide at such a big thrust of factor IX going through it. You can tabulate that. You can look at the values of activity. And when there's furin present, you don't see any propeptide when you quantify it. Look at the difference in the activity. So these are clotting activities. This is a single-stage clotting assay. And the monogenics that just have factor IX being expressed at the same levels, about 2 grams per liter, as the bigenic mice under the same genetic controls, that's really good, they have half the activity. The theoretical activity is around 200. That lower activity is due to the propeptide. It can't clot if it still has, it can't catalyze clotting if the factor IX still has the propeptide on it. We could take the monogenic milk and actually mix it into the milk made by the bigenics. There's enough furin to clip that off too, and you see the activity goes up. So it was a really nice example of robustness. Now when we purify this, and we purify to an endotoxin-free level, which for academics is a little bit more difficult. It's hard to explain to a student why it has to be that way. But I'd say, look, if you don't want to give someone a really bad fever, a dog that you're interested in as a pet, don't do that. Make sure the endotoxin is out of it. So they're motivated. This isn't a hemophilic dog. So these are hemophilic type B dogs. They don't make any factor IX. These are at UNC Chapel Hill under the direction of Dr. Tim Nichols. And when we go with crossover infusions, these are IV infusions into these dogs to see what kind of activity and how long it lasts. Don't forget, these are facsimiles that we're making of factor IX. So the ability to do the clotting, we verified that in the test tube from an enzyme point of view. But we don't know that complexity for how long it's going to be in circulation. How is it going to partition? How is it going to partition that's something you can only do in a living creature, and you have to do it in a large animal. So you have to have a lot of factor IX to do that. That's what I was talking about before. Whatever you do to pioneer these new, in this case, factor VIII and factor IX molecules that we're talking about tonight, you got to make a lot of it. Otherwise, you really can't characterize, even at the research level, you can't characterize what's going on with it. And so if we look at crossover studies, and you see two different ones where we put the transgenic first and plasma-derived second, or plasma-derived factor IX and the transgenic second, so we can see differences in how long it lasts and what kind of clotting behavior. So our pharmacokinetics here track the clotting levels. The lines in the middle are basically the normal limits, plus or minus, that like all people, where normal clotting occurs in the dog. And you see that we're able to sustain it with these infusions, no matter what comes first, for a very long period of time. And it turns out 40-hour versus 22-hour pharmacokinetics for the factor IX molecule that's recombinant. That's a good result. If we look at, and we don't have a IACUC approved procedure for assessing bleeding, cutting, and looking at blood accumulated with a gauze, or that we don't have that in a dog, so we have to go back to the mouse and look at that. So we do that kind of an assay in a mouse where we can get permission to cut a tail. You know, 3 blind mice, right? The tail gets cut off. It's not a bad thing. The clotting activity, the clotting activity of the currently approved product that comes from Chinese hamster ovary cells versus the transgenic factor IX, it's equivalent, both in the initial clotting that we see in blood loss versus the wild type and the hemophilic mouse alone. This is injected into hemophilic mice. So they're knockout mice. Their factor IX gene has been knocked out. And then the rebleed behavior, which is a very important phenomenon in hemophilia. The rebleed 30 minutes later in mice is a good model for how is the factor IX, what does it do in terms of generating stability in its hemostasis over time. That's a good assay for that, and it's equivalent. So from what we can tell on a preclinical basis, I think we have a reasonable case to say that someone should do this in a human clinical study, provided now a bona fide biopharmaceutical company can take this up. We think that's more likely to happen in a developing country than it is in the competitive market in the United States. After all, if you invested $1 billion just a couple years before in a factory to make that recombinant factor IX I showed you up there, that's approved, what is your incentive to find a new one? You got a long time before you make that $1 billion upfront investment back. This is a problem. This is a problem. So even though you have something that has really favorable pharmacokinetics, favorable hemostatic behavior, how do you convince in a place, in a country, for example, that has everything, that's already made their investment, how do you convince them to go after that? Very hard, particularly if you're an academic. So what we look at is if we use these pigs with the kind of yields that we see from that chromatographic processing that you would use at large scale, just like they do to make the recombinant factor IX that we used in that bleeding assay as a comparator. With the current level of just fixing the furin, you're only talking about 2,500 milking pigs. Now I realize there's not much of a market for pig cheese, that they're going to have to be dedicated to this. But I would say that, for example, Smithfield in Virginia kills 15,000 pigs a day. 2,500 pigs for the whole world market. That whole map that I showed you, it's not much. If we make the next modification just to up the carboxylation a little bit, because it is at a rate limitation for carboxylation, not all the factor IX is carboxylated, then you have just a handful of pigs, 900 pigs. That's a gentleman farmer. To make enough milk to purify, to treat every hemophilic B patient on the planet, including us, 900 pigs. That's what the tissue structure of the mammary gland can do for you if you can genetically engineer it. It's made to pump out protein, and complex ones at that. What's more, this is one of the most fascinating things. Long ago, I met a hematologist from Holland named Hemker. Dr. Hemker did a really interesting experiment. Some fractionator in Europe gave him a little bit of factor VIII. And we were talking at dinner tonight about how sometimes you just have to do experiments that you have a hunch for, that it has some merit that's maybe not that an NIH grant will be granted from it, particularly in today's current situation. But it seems like it might be, if something happened and came together where we could do that, it'd be great. So Dr. Hemker formulated this with a lipophilic, a soap bubble type of packaging, factor VIII, and gave it to his most severe hemophilic patients. Just a couple of them, because he didn't have much. Didn't have much factor VIII. And guess what he saw in their blood? He saw clotting levels that were above 5% and essentially rendering them normal for long periods of time. When you put it intravenously, it falls off quickly. Look at the profile of that rise. It rises in the middle, plateaus, and comes back down. That's because it's taking a path, an enteric path, either buccally or somewhere along the line, enough of it protected from proteolysis, from hydrolysis in the stomach by stomach acid, to get into the bloodstream and render essentially a normal phenotype for a good number of hours with a different pharmacokinetic behavior because of that lag of getting in and then eventually getting into the blood. Remember that graph I gave, or that figure I gave you about extravascular storage space and what's in blood and in balance with what's in the extravascular space? This is what happens with blood proteins. So the pathway it took to get into the blood is very curious. But it said, boy, that was a great idea. Could we do that? We've got a whole bunch of factor IX. Let's feed it to some hemophilic mice. And we did this at NIH. We didn't get high levels because we didn't have any ability to formulate it in a way that people with buccal delivery experience and enteric delivery experience to optimize that kind of formulation. We just fed them the milk. Turkish milk's not bad. We got detectable activity, and we were shocked. Hemker was right. He did it with factor VIII. We did it with factor IX because we had enough. So then we did some experiments with hemophilic dogs and saw just a tiny, tiny amount, much smaller than that. And we said, wow, not so good in the dogs. That's the penultimate preclinical animal, large animal to humans. What's going on? We did it in normal dogs. Normal dogs have the reservoir full already. The pathway this is going to take, buccally or enterically, will be curious. What factor IX are we going to be measuring if we see any in the bloodstream? Maybe stuff that went inside out into the reservoir and not into the plasma, but kicked the factor IX that was in the reservoir out into the plasma, and that's what we're seeing. And in fact, that's what we saw. So after just an hour or two after feeding these dogs this milk, we saw a rise to 20% above normal of the antigen, of the protein itself. We saw the same thing with activity. If you have a lot of material and you have an amazing technology, you may be able to do more than just baseline improvements in quality of care. You may be able to end IV therapy for a good part of the time for a lot of patients. And you have hope with that, with this kind of experiments showing that. So Hemker was right. So this whole idea of having lots of material, being able to do large animal trials, studies, allows you to understand a lot more things that you couldn't by just infusing into a mouse at the few micrograms that you have. That was a big lesson. Let's talk about factor VIII. Just for a little while. This is a scary molecule. The first time I started working with factor VIII, I met a very, very famous French scientist. And he said, "Bill, why do you want to do this to yourself? Factor VIII will destroy you." He spent his whole life on factor VIII. And, you know, he was right, except that I had a whole bunch of really good factor VIII people standing shoulder to shoulder with me as a team to work on factor VIII. I didn't explain that to him, but I got the message. So factor VIII is not to be trifled with. It is a huge molecule, 300-something thousand Daltons. It has a very complex relationship with VWF, which is 440 kilodaltons. And so two really complex things forming a complex. And then trying to figure out activity based on the fact that it has to do that. If you don't have VWF in your plasma, you will behave like a type A hemophilic person as if you didn't have any factor VIII. Because if it's not packaged with VWF, the proteases in your blood will destroy any factor VIII that your liver secretes into it. So very complex problem to solve. You have to have all of this other things going on to be able to understand, are you making a factor VIII that is stable and is active? That's a tough question. So this is the one. This came to me as a JPEG called Smiling Piglet. And it was really a great thing because we were in competition with Dolly the sheep for this particular edition. And the people at National Geographic were so nice. They had 3 small kids that came to Virginia Tech, and we introduced them to our farms there. And they focused on our Smiling Piglet. So all I can say about this is we're grateful to Washington. We're grateful for the National Geographic Society. And Dolly the sheep got just a picture that big. We got 2 full pages plus the cover. Pigs rule, sheep drool. The real story here, though, the real story that we got this is because we got a great Nature paper in this expression, our first try at making factor IX. We made some, but it turned out to be a deep, deep puzzle, although we did make some progress. All of the chains, factor VIII in your blood is chopped up in little segments. They all hold together if it's going to be active. And we had in blood versus what we saw in the milk, we didn't make that much in the milk in this first try, but it looked like it had all the right parts. That was an SDS page Western. And so you're looking at just the antigen. You don't see activity in that. But when we looked at the activities, we were a little more depressed. On the one hand, the 10 to the 9th cells per ml of milk that are in the mammary gland, in spite of not making very much factor VIII, factor VIII is absolutely restricted at the transcription level. It does not want to be transcribed. Your liver cells make only 200 nanograms per ml because it's restricted. It can't do a good job, and it does the best job. So, you know, again, it will destroy you. So to that, I remember that when I saw those units per ml. However, if you add it up to the same bioreactor levels, we did get 12 units per ml per every 24 hours. The bioreactor only made 0.75 units per ml per 24 hours. That's because they don't have many cells in their bioreactor, and that's okay. But we are still wondering, we had all the right parts there. Why wasn't is it as active as it should be? It's specific activity, you see this, but only 0.5 to 0.05 units per mg, units of activity per every, microgram of factor VIII made, where in this case 5 would be the normalized if it were perfect. So why so low? We made a lot of factor VIII, and it's all there in the right pieces, but it's not assembled somehow. What do we need to do? We need to re-engineer it. I talked about before that you have to, with these kinds of molecules, it's not only a facsimile problem, but you may have to go to the point where you just redesign the molecule to make it a doable thing in a bioreactor. With factor VIII, that's what most of the industry does. So we worked with Steve Pipe and Randy Kaufman at University of Michigan, who were Randy was the first one ever to make a recombinant factor VIII product. And so he's just one of the pioneers in factor VIII, recombinant factor VIII, and Steve was one of his understudies. So we had the best people in the world working on this. And we came to the point where we say, we are not going to allow this thing to fall apart. We're going to have a single chain. We're not going to allow it to be cut like that. So we redesigned, with their help, that molecule and put it under the control of the promoters that I talked about before. But we did one more thing. That's Dolly the sheep and her progeny. I don't have anything against Dolly the sheep. She was a great animal to pet, and counter to really what most people say about her demise. She's on casters and stuffed in the Edinburgh Museum. She had a great life. One of my PhD students palled around with the PhD student that made Dolly as just a graduate student process, just a product that was on a whim. Another thing seemed like a good idea at the time, and it worked. But we used the gene that was put into her because it was one of the most highly optimized milk-directing, lactation-specific genes ever made. It made 35 grams per liter of alpha-1 antitrypsin. Dolly the sheep made alpha-1 antitrypsin, which is something that could be very helpful for cystic fibrosis because it stops degradation of lung by plasmin from neutrophils that would essentially the immune inflammation secretes this plasmin, and it needs to be stopped. The antiplasmin that was made in her milk could help that. Now, Dolly wasn't for long, but it wasn't because of her short telomeres. It was because the business decisions were bad, and two companies got in a war with each other. So everything about science, and amazing science like Dolly the sheep and cloning, has a dark side that doesn't even come to light, even if you have enough NIH funding. The remaining step, the valley of death, the getting through to actually make the products, is still a big problem for biopharmaceuticals and all the miracles that they should be able to bring, that they promise and should be able to bring to everyone. That's the lesson from Dolly the sheep that I think is important. But we used her gene, which is very highly optimized, and that gene, if it could be co-integrated in the same place as the factor VIII and the VWF gene, so we got 3 genes in there, could that make a domain that would be more responsive, that the chromosome would unwind at that point, that the gene would then transcribe, even though it doesn't want to because it's factor VIII? John Clark, who made all the things that have to do with Dolly the sheep and making alpha-1 antitrypsin, spent 10 years of his life optimizing this gene so that it could do things like that. The theme is gene rescue, a gene that just resists being shut off, that wants to be transcribed, that can make a domain that will be transcribed. If we co-integrate into that domain, then maybe we can coax factor VIII to be made. And in fact, that's what we saw. We saw it both with VWF, that's the bottom line, so it's an SDS page that is a Western blot showing the VWF antigen in the milk. And the top one, you can see reference factor VIII and the recombinant. How big was that? That single chain nature produced a specific activity now that was very close to theoretical. The 5,000, if it's milligrams per milligram, the 5 if it's per microgram, that was a key. The redesign of the factor VIII and even getting it into the milk because we did gene rescue, we made it, we did not say no for it couldn't take no for an answer to be expressed because that BLG gene was there. And that was connected to Dolly sheep, and I think that's an important thing about if you were to get the most tough problem solved, you have to collaborate with some of the smartest people in the world. And I am really glad about that kind of blessing I've had in my scientific career. Having smart people around you. And my mother told me that. Work with smart people. She was right. It works out good. So that factor VIII that was made in the milk is exactly comparable in a bleeding assay. I showed you a bleeding assay in mice before. This is a hemophilic A mouse. It's equivalent to what is made by CHO cells. That is the original project from Randy Kaufman, by the way, but is now marketed. It's approved. It's what most hemophilic A patients take. So we said that redesign worked. If you look at all the past examples of people who tried to make factor VIII that are most comparable to what we're talking about, the only ones I don't include are the ones that were absolutely zero. So it's not worth talking about. But you see the amount of activity in units per ml. Look at the difference. Look how many units per ml. Look at the concentration of these. We had mice that expressed as much as 300 micrograms per ml of fully functional factor VIII, up around 4,000, as is the rest of them in the reference cases made in the bioreactor, stainless steel bioreactor. So I think by using this transgene rescue theme and by having VWF around so that it would complex and be protected from the milk proteases, milk is a very proteolytic environment. That's one of the downsides of this. We were able to avert that. Okay, what does it look like in terms of number of pigs that would take to do this? It's about 40,000 liters of pig milk to do the whole world. That does not get, they don't get factor IX, they don't get factor VIII. So if you look at that, for the stainless steel bioreactors with the Chinese hamster ovary cells in it that are making factor VIII, look at the difference in the volume of bioreactor fluid that you'd have to purify. That is the advantage of doing it in milk. So where are we at now? We hope to make even more. We're going to use gene editing. We know that our factor IX genes, for example, create also very favorable domains once they're inserted. They make lots of factor IX, grams per liter of factor IX, not hundreds of micrograms per ml of factor IX, but grams per liter of factor IX, that's a lot. What if we took gene editing, which just recently became available, and inserted into those sites the factor VIII genes plus the BLG rescue? What could we do to really push that? Here, this is a fish assay from one of our transgenic pigs, and you can see the glowing stain there. That's the insertion site that we want to go into with a factor IX. We'd like to insert in there the factor VIII, BLG, the VWF, go in there using CRISPR-Cas9. Just, I think, a kind of a miracle type of gene editing process. And if we can use that to insert in that, that's our next step. In the meantime, the people that are supporting this is the Ministry of Health of Brazil. They can't afford a $500 million a year of importing factor VIII. They can't afford a $1 billion factory to make factor VIII. So they have some hope in this. I'm glad they see some of the rationale, and that's great. So the only thing I want to talk about now, I talked about cloned pigs. I'd like to talk about cloned cows for just a minute or two. And this is, again, making a very complex protein. It shifts the scale. It shifts the scale. We're talking about if we treat the whole, by IV, for example, the whole world with factor VIII, we're talking just kilograms, just a few kilograms of factor VIII, because it's very powerful. It's powerful at 200 nanograms per ml. Factor IX is 4 micrograms per ml at normal levels. Not much. A few more kilograms. Let's talk about a protein that we need to treat surgical wounds, to treat trauma from car accidents and farm accidents and violence. Let's talk about what we'd need for a recombinant protein that's not quite as complex as factor VIII or factor IX, but in the top 10. And you need a metric ton of it per year. What could you do with that? You got to find an animal that can make it, but also make a metric ton per year, at least a herd of around 400 or 500 animals. How about these bioreactors? They're some of the, I think, the cuddliest, cutest bioreactors I've ever made. Those are clone cows. Those are Swiss Browns. They make about 90% of what a Holstein will make. I was brought up in the Illinois-Wisconsin border, so everything around us was Holsteins. But these are not shabby. 90% of what a Holstein makes is a good milking cow. So we cloned fibrinogen, the thing that is the barrier, the hemostatic barrier that I said is the ultimate product of the coagulation cascade. Remember a long time ago at the beginning of the lecture? It's a pretty complex protein. It's got 6 separate chains. It weighs 340 kilodaltons. It's a very, very big, complex molecule. But it has to be able to form that polymer instantly within 3 minutes to stop bleeding. It takes 3 genes to make 2, 2, and 2 of alpha, beta, and gamma and make a hexameric protein. The question is, can the cow do it? So this is from, this is a cow, one of our first clone cows named Fancy. Fancy that. Fancy that it was able to happen. She made about the same amount of fibrinogen as is found in human blood. And it's almost identical with a couple of small twists. And those twists have to do with post-translational modification, as one might think or expect after seeing factor VIII and factor IX and all the things that happen with that. It has to be made into a polymer. It's made into a polymer by thrombin, which causes it to aggregate and interlock with itself. And then factor 13 locks it together. So it's a very you're making a tire patch, but it's not a simple tire patch. If you look at the way it works, if you don't have factor 13, and so we had to make recombinant factor 13-2, we made that in yeast because it's not so complicated. But if you just activate it with thrombin, that clot will dissolve if you just put some soap in it, SDS in this case. But if you put the 13 in it, you form a nice stable clot, cross-linked, covalently cross-linked. This is a good sign. This is a good fibrinogen. And it's got some differences. It doesn't have the same glycosylation. Glycosylation actually makes a difference if you're trying to make that fiber structure I just talked about. This is the equivalent it's far enough past post-dinner, and I talked to people, mostly in the audience, about what they thought about Julia Childs, and they all agreed, save the liver. So I'm saving the liver. We're putting this recombinant fibrinogen on the wound. This is a standardized wound that's used to develop hemostatic devices, things that will stop horrific bleeding from things like car accidents or shotgun wounds. This would be very much like a shotgun wound to the gut. But you see, within about 15 seconds, that the fibrinogen, this is all recombinant thrombin, recombinant factor 13, and the fibrinogen isolated from those Swiss brown cow, genetically engineered cows. It's like caulk. It's like caulk. And it's because the glycosylation, the post-translational modification, allows these strands to form in much tighter bundles. And they're incredibly wound adherent. Oops. If you look at a normal clot, it's very diffuse. So if I look at a wound surface like what I showed you, so a histology taken from that previous slide, you see it's a very diffuse clot on the surface. The clot formed on this pig liver on the surface is very dense, That makes sense based on that white caulk picture that we saw. Okay, so where are we at? We've made a bunch of different proteins in these bioreactors. Now, the pig isn't as adorable as a bioreactor like the cow, but as piglets they are, but not when they get bigger. They're just a bioreactor. But they do make significant amounts of material. And if I look at this cost thing I was talking about before, that's probably the only thing I can say about chemical engineering. I'm in a chemical engineering department. That really was a good thing. We can make these calculations in this kind of a department. And what you see is that the production cost, this is the other cost. The biggest cost is the $1 billion up front to make the factory. It's only 1/10 as much if you're using a transgenic animal to make your starting material from which to purify. Once you purify it, the cost has to do with what the scale is. If you have people standing around because you've got a certain scale operation, it's going to cost you a lot of money, and that's fixed on a per gram basis. But if you're going to make a metric ton, like that 1,000, like the cows can do, then you can get it down to less than $1 per gram, which is really cheap. So the production scale economics work. The fact that you don't have to invest up front works. You have a small herd of cows to be able to do this. A gentleman farmer's amount of cows to make a metric ton of fibrinogen. But you still got to deal with this. That's the next stuff. The next level has got to be brought forward through the valley of death. Here's factor IX and factor VIII. In fact, fibrinogen, this is where we've taken them. To get through the valley of death, there's a significant amount of money that's going to have to be done, and it has to be done by professionals who do this. It won't be done in the United States. The French might do it. In Europe, we have some good feelings about that. But the developing countries will have to do it themselves. And perhaps that's the best thing. I'm happy to take questions. I appreciate you staying awake.
Bring you the microphone. When you get the microphone, please stand up, tell us your name, if you're a member of the society or not, and then ask a question. So, who has a question? Please put your hands up in the air. There's a question up here. Carl?
Carl Merrill, a member of the Society. I really appreciated your presentation, a tremendous amount of work. I remember being fairly old. During the Second World War, of course, there was a great concern for wound bleeding and during surgery. And they didn't have any of this knowledge, so they couldn't even approach it this way. But what they were looking for is material from snake venom and that sort of thing. And I wondered, has that progressed at all? Or is that just totally...
So that's a really interesting question, because if you go back and look at that coagulation cascade and you look at the penultimate steps for generating this hemostatic barrier, thrombin comes out of this coagulation cascade, and it's the chief catalyst for polymerizing the fibrin. Is there enough endogenous fibrinogen at a wound site for the thrombin to act all by itself? For many exsanguinating level wounds, probably not. And so it's used for, say, nuisance wounds that happen in surgery, and you can sprinkle it on. It's very good at that. So that's where it is good. So it's not a, it's a gradation question, but with insanguinating injuries, probably not. With the way it can be introduced by spray or by particles that contain the thrombin on it, probably not. It's a great thought for nuisance wounds, nuisance bleeding and surgery. It works quite well. Great question, though. Great question. Because if I wish it would, because it would simplify the whole problem and cost everything. And we can make we've made prothrombin in pig milk before, so we could make thrombin easily. Good question.
Stuart Umpleby, I'm a member of the Society. This has been fascinating. I didn't know about all this work going on. I know that there's been a lot of concern about genetic modification, particularly in Europe with crops and things of that kind.
Yeah.
After you've produced a pig or a cow that produces the factor VIII or factor IX. Is that milk dangerous to humans?
Or do people perceive it as dangerous to humans? That's probably
That's
an even
fine.
bigger one.
But, I mean, does that animal then pose a risk if interbred with normal animals and that kind of question?
Yeah, that's a really good question. And, you know, when we first...
Candidatos, no se quede afuera en la carrera presidencial. Siga al equipo
Does
más
that answer?
profesional en la más extensa cobertura de la...
Yeah, I said, gosh, my Spanish has improved tremendously. What's going on? But I got to shut it off. This is what I would say. When we first started this project, the GMO, anti-GMO activity, particularly with animals, was a big, and I would say legitimate, because it was totally a new world, a brave new world. And so we spent a lot of our time being proactive, giving talks everywhere we could. The Pew Foundation did a beautiful job with us and sponsored a lot of our work all over to make sure it would get on. And we could explain answers to questions like this. And the simplest answer is, this is in milk, for one thing. And its proteins in the animal are very similar because they're mammals. They have the same coagulation system, although their proteins are a little different. But we have to make sure that our purification doesn't contaminate the endogenous protein, bovine fibrinogen, into the human recombinant, pig factor VIII into the recombinant factor VIII product, and factor IX, et cetera. And so they're a little bit dissimilar. But if you cut your finger, which everybody does, and you bleed on the pavement like everybody does, you've contributed more of a problem with those proteins spilling into the environment than these animals are going to make. So the fact is those proteins are more or less already there. We've just fine-tuned it a little bit to be a little bit humanized. They're facsimiles of human, so they're not perfectly humanized versions. And so there still is worry. So what did they ended up? What is the precautions? Every one of these animals is when their lifespan is due to be done after serving a very noble role, they're incinerated, and so they're not buried, they're incinerated. So do I think they need to be incinerated? No, I think they need to be eaten, because it's not a problem. And we tried to argue that, and the FDA says it's no problem. You just have to label everything as genetically engineered so that the people who buy it know it, and they make the choice then. They don't have to buy it if they don't want to, but it was clear from the FDA's point of view, and we got proactive with the FDA from the very beginning, and they got proactive with us. We held a meeting every year from the beginning since 1987 for 8 consecutive years, where the FDA was at this meeting for 4 days up in the mountains in Virginia, where we just talked about these things with a very open collar sort of discussion, saying this is what we think the dangers are, or lack of dangers is for these reasons. And so they said, okay, we'll write the points to consider for these proteins being expressed in animals. And it turned out to be scientifically very, very well based. And so labeling became the biggest deal. The fear of the animals getting out, they're in restricted access facilities. They're not likely to get out. If they do get out, it's really, they have a very human-like protein, a very animal-like protein in them that's very similar to the one they have. So, and it's only going to be in milk. It's not, it doesn't go anywhere else. It isn't made in any other tissue. So that confines the kind of the threat in a lot of different respects. Excellent question, a tough one to deal for the first 5 years. Thank goodness everybody got together, scientists from all over the world, the PPL, Dolly the sheep people in Edinburgh, the people in Holland. You mentioned Europe. Yes, they were very rattled by that too. But we were very proactive, and we discussed these things in a very open way. And ultimately, they all agreed. All of the people, even Jeremy Rifkin agreed, that the risk, which was unbelievable, that the benefit to risk ratio in this particular case was dramatic. He didn't say that, oh, you're done with us. He said, we're going to watch you, because maybe it'll turn into something bad. But he agreed that the severity of the diseases that the severe nature of these diseases, that what we could bring to treating them, he said, that's got a ratio that's probably justifiable. So even he did that. And that was a very unnerving point-counterpoint. We had that on CNN. But I have to say, I said, I am not, I'm going to show you some videos of kids that have these diseases that can't get access to these therapies because they are so expensive. And he looked at that video, and he came to the conclusion at the end of this point-counterpoint show that, yes, indeed, the risk-to-benefit ratio is astonishing here. So, but we'll still watch you.
Bob?
But it was proactive, dedicated to proactive conversation, dialogue that got us to a point where I think most people were comfortable.
Bob?
I'm Bob Hershey, I'm a member. What do the time periods look like from here on?
That's a good question. I think the fibrinogen project could go into clinical study now. I've convinced the French government, who have their own biopharmaceutical company called LFB. Still, the mandate for LFB is to make medicines available for the French people and then sell it everywhere else and make money. But they have agreed to take these cows over and try to commercialize them. So that one, I think, will go within the next 5 years into clinical trial, because it is, as far as we can tell, really good fibrinogen from really high-level, large animals, preclinical study. Factor VIII and Factor IX, a little farther to go, but the Brazilian government just took up that charge for Factor VIII, and I think that they will make it go within the next 5 years. Factor IX is less of an urgent need, only 1 out of every 23,000 kids, males, born. So it's not many people urgently wanting to pursue that, but Factor VIII is a 1 out of every 5,000 born. It is a big deal for Brazil. Fibrinogen is a big deal for all sorts of exsanguinating trauma. And so people want to take the charge of that. So I'm hopeful in the next 5 years something's going to be in clinical trials in humans.
Next question. Just speak into it.
Elisa
Your name?
Nguyen, I'm a guest of a member. What other, I realize that oral delivery in milk probably limits it, but what other sorts of things are people thinking about now that you've developed this process?
Well, you know, that's an excellent question, and the abundance, we have spent so much time on just being careful about qualifying what kind of abundance are we talking about, what's the quality of this very abundant recombinant Factor VIII, Factor IX fibrinogen. And in the case of Factor IX and Factor VIII, the oral delivery would solve a huge amount of problems in the developing countries because they can't even afford a technician or a nurse, someone who's skilled in administering IV healthcare. It's a tough thing. So even if you have it, they can't afford the person to infuse it. And so can you imagine being able to just reconstitute, you know, with some bottled water and drink it and be able to have some level of normal levels of coagulation? How much of a miracle that would be? We think that's where everybody wants to go with this because that solves a very key problem. What else could it do? Our pharmacokinetic studies in dogs suggest that, and we're trying to get some support to schedule the next set of dogs to be able to prove this concept, but that probably a good question to ask is, joints don't degrade over a lifetime normally because you have constant levels in blood and you have constant levels in the reservoirs. If you have an abundance of this and you can adjust that equilibrium so it doesn't fall off so much that it kind of goes maybe like that, and the levels remain full in the extravascular, what we have seen with some of our probe runs is that if, and this gets back to this omega loop thing that I didn't have a time to talk about, is that you can control what is filling the reservoirs and therefore what can't get into the reservoirs once those reservoirs are full. Because you get a very stable filling, and the transgenic will do that for you. And if you inject the ones that are now clinically approved, after you've filled it with the transgenic, what you see is you dramatically extend the residence time that's normally only around 22 hours for these already approved factor VIII and factor IXs. You extend it to like 40 because it can't get into the reservoir because of the preferential avidity due to this omega loop thing I didn't get a chance to talk about, because that's what locks the reservoir and unlocks the reservoir. And so I think that's going to be a big thing for people to investigate, because that would change the way we treat people here in the United States. We would see this effect, because we have a lot of it, you see this effect of extended plasma life while having extended reservoir filling. What does that do for the condition of joints if you maintain that? Because that's what we do normally. For normal people that aren't hemophilic, that's what we have. So if you could engineer something that's closer to that, how does that affect joints? Understanding the physiology of the disease effectively, because you have a lot of material that has not facsimile, not exact copy of things, it's a facsimile, but it has uniquely different things that may be a useful tool to understanding the disease. So that's where I think that's going to go. That's a good question. It's a deep one, but and we would need a lot more NIH money to be able to go after that. I don't want to put any more plugs in for NIH money. That's a hard thing. I'm sorry for that.
Hi, this is Jane. This is Jane Tian. I've been working on also like transgenic animals. Like for some plasma protein, such as albumin, the annual consuming, the amount of consuming amount is like more than 500 metric tons. And for that kind of protein, if you're trying to get through milk, there's lots of problems. Like if the concentration is too high, and the milk will cease to produce. Now the strategy I'm trying to make is just simply using that CRISPR technology, humanize that pig gene, and then in the blot there will be human albumin to replace the pig one. So I would like to have your general comment, this kind of.
Yeah. So that's a really on the one hand, it's a very perceptive way to address contaminating proteins into the milk that would be detrimental, that would be not easy to separate out from your product. That's a really, what you're saying is a very good thing. Now, because whatever passes over from serum into the milk would be more or less an equal human facsimile to what you're making in the mammary gland. With albumin, that would be very true, because albumin is not glycosylated, and so doesn't face that kind of fingerprint that the liver would put on it versus the mammary gland. They put different fingerprints in terms of glycosylation. So, having said that, we looked at albumin a long time ago, because it was a very, very big, 50% of the revenues from blood fractionation in the world were coming from albumin. That has changed, and I'm going to tell you why. It's a surprising thing that all of the theory about why albumin is good for you, for example, osmotic regulation, so that if you've had exsanguination, they fill you up with saline, you don't have the proper osmotic regulation because you don't have the albumin there. That was one of the driving forces. So fill up with saline and also fill up with reconstitute with albumin and get a much more stable recovery from the near exsanguinating event. It turns out that that's not true, that it doesn't really do that. And in addition to the fact that it has some other paradoxical problems when you put that much albumin in. And so now the amount of albumin is precipitously dropping, and the only country that really believes in it, stubbornly I might add from my point of view, is Japan. Japan still uses a huge amount of albumin, whereas everybody else has backed off tremendously from using it. So when we first got into this, yes, albumin was one of the top things because it was such an impossible amount, and it was important to every developed country. It was a standard therapy for reconstituting plasma to levels that were near normal. And it turned out that none of those things were really beneficial. And so Japan's the only one that actually believes that, and they're starting to cut back. But still a fabulous question, because at its root is, if you don't want animal contaminating versions of those proteins coming into your product because you're purifying for a specific thing, and the thing that the animal has endogenously, natively, is very similar to what you're trying to purify from the milk. That's where I think your question is fabulous. Thank you.
Last question, David?
Okay. You said that you implied in one of your, that most of the people with hemophilia, at least this type of hemophilia, were de novo mutations, that they didn't breed, that the rate of new people with hemoglobin was actually the mutation rate, was determined by the mutation. What if you undo that and all of a sudden people can breed, who aren't being killed so much, wouldn't there be more?
Yeah, exactly. That's a really good point, is that if you cure by restorative therapy of the factor that's deficient, and then the people, actually studies was done on this, exactly this, showing what happens if they're healthier and they're able to have more children. Do you further propagate in an advanced rate the incidence of hemophilia? That's a very good point. And they did say that more healthy hemophilic patients did have more children and did have more incidence of hemophilia that came just from that. So that would be on top of the endogenous frequency that's going to happen no matter if that happens or not. So that's a great question. And yes, there are studies that have shown that would be a problem. So the next logical thing is to talk about gene therapy and stopping that. Gene therapy, gosh, I hope it happens. I work with the guys at UNC Chapel who have developed the AAV vectors. There's a whole bunch of serotypes now of different adeno-assisted vectors that can specifically target different tissues and allow a stable transformation from a gene insertion point of view, but not necessarily a gene expression point of view for a long time. So there's the complexity in that. But it brings one more thing that is exactly what you're talking about. They're devising all the gene therapy to not be germline, to avoid curing, transfecting the testicles, for example, so you would have a hemophilic person that would not transmit that. So it goes exactly the opposite of what we're talking about here, but that's what gene therapy would do right now if it given a choice. They would still have the defect to be transmitted genetically. So it has to be talked about when gene therapy gets just a little bit better than it is right now. But great question, though. Great question.
As the speaker last year said, biotechnology is the technology of the 21st century, and I don't think we're going to settle all the questions tonight. But we are going to present you with a framed copy of the announcement of your talk in appreciation for your giving the lecture tonight, signed by the members of the General Committee on behalf of the membership of PSW. So with that, thank you very much.
I'm greatly honored. I was greatly honored. Thank you.
This is
Thank you.
Off the stage and wait a minute.
Yep.
So before we go, there are a few housekeeping announcements as usual. As many of you know, PSW depends on an active, enthusiastic, capable membership. Without these, there's no PSW, no free lectures, and we don't get to listen to people like Bill. So please join if you're not a member, and if you are, get involved in helping carry out PSW activities. In particular, we're looking for members interested in helping us with writing, media and web development, membership development, grant writing, and publicity. If you have skills in any of these areas and would like to explore volunteering to work with PSW, please see me after we adjourn to the social hour. You can apply for membership if you're not a member very easily on the PSW website. Believe it or not, there's a little button that says membership. And you click on that button, you fill out the form, a dues payment will come up, and you're done. It's easy. Just go to the website. If you're a meetup member, please keep in mind that being a meetup member, while it's wonderful, does not mean you're a member of PSW. So please consider becoming a PSW member and contributing to the organization. Please see membership chair James Heelan. Don't see him because he's not here tonight. Our corresponding secretary, Robin Taylor, or our vice president, Lloyd Mitchell. If you have any questions about membership, if you're eager to give us a contribution, or if you would like to get involved. PSW is a nonprofit educational organization, tax exempt under Section 501c3 of the Internal Revenue Code, and contributions to PSW are tax deductible. Our next lecture, the 2361st, will be on April 1st, 2016, not an April Fool's joke. The speaker will be Stephen M. Kahn, Kirk Professor of Physics and Director of the Large Synoptic Survey Telescope, Stanford University. He will be speaking about surveying the universe in high definition, the Large Synoptic Survey Telescope. It's a pretty amazing instrument, and I think it'll be a very interesting talk. Finally, the spring schedule has been posted to the website. There's almost certainly going to be a change in the schedule, and the Henry dinner, instead of being the last dinner of the year, will be the next to the last dinner of the year, and the Henry speaker will be Alan Stern. Alan Stern is a co-author on 5 major papers that just appeared in Science, reporting the results of the New Horizons robotic exploration of Pluto and the Pluto system. And this should be a pretty fabulous talk, and I urge you all to come. The social hour ends at 10:30, after which PSW members and their guests meet at the Fairfax Hotel Lounge just across Massachusetts Avenue. If you want to join us, but you're not sure how to get there, please look for Robin Taylor, myself, or Lloyd Mitchell, and we'll be happy to invite you. You're welcome to be a guest on a few occasions, even if you're not a member. I will now accept a motion for adjournment of the 2360th meeting of the Society to the social hour. A second? All members in favor? All members opposed? We are adjourned to the social hour.